Hi everyone, in this video we are going to be talking about mutations. And to start us off, I just want us to think about this question, which is written on this slide. And the question is, how do new traits arise? So when we're thinking about a new trait, so maybe we're thinking about a new color of something, maybe a different shape that something could take, maybe it's an enzyme that gets a new function, any sort of new trait, how does that come about? And for the past week or so, we've been talking about protein synthesis and how we go from genotype to phenotype. And the way that that happens is because our DNA contains the information for how to build different kinds of proteins. So if we're thinking about different phenotypes or new traits, then hopefully we can sort of reason backwards that those new phenotypes, new traits, have to be the result of different kinds of DNA instructions. So, if we're trying to get different kinds of DNA instructions, we have to change the DNA in some way in order to make that happen. So, this is where mutations are going to come in. A mutation is any change in the nucleotide sequence of the DNA itself. And it can be just a tiny little change, so a single nucleotide can be altered, or you can have differences that affect the entire chromosome. So we'll be focusing more on mutations that are on the smaller end of this range, but I do think it's good to mention that they can be different kinds of sizes. Mutations are relatively rare. When you think about it, it's actually kind of wild that you have so much DNA in every single one of your cells. You've got those 46 chromosomes in every single one of your cells, but mutations aren't causing us problems left and right. They're pretty rare, all things considered, and that's because your cells have specialized machinery to help keep the mutation rate down at a pretty low level. Okay, so the next bit of this presentation, we'll just be talking about the different kinds of mutations that we can have. There are a few different types, and they're listed on this slide. So one type is called a substitution or a point mutation. And the point part of point mutation just gets at the fact that it's a single nucleotide that's affected for these types of mutations. So we're exchanging just one single nucleotide for a different nucleotide. So maybe in this example, we have a T at this position right here, and we're changing it to a C. So going from a T to a C. These mutations, since they're so small, can actually be silent. And we'll talk more on the next slide about what that means. But basically, some of these point mutations don't actually have any effect. It's basically like there's a mutation there, but it doesn't actually make any difference. And we'll talk about why that can be in a second. But the other two types of mutations that we'll talk about are called insertions and deletions. And in insertions and deletions, we're either adding, in the case of insertions, or getting rid of one or more than one nucleotide. So in this example here, we're adding in a C in between. So here we have a C next to a T, and here's that same C and that same T, but we've added in a C in between. And it can be one, it can be a lot. You can have insertions and deletions that are different sizes. So these are showing just one, but here in between, or not in between, but for that C and T that we were just talking about, now the T is actually going away. And the reason that insertions and deletions can be so catastrophic is that they can result in what are called frame shifts. And I know I'm throwing a lot of vocabulary at you on this slide, so we'll talk about each of these in a little bit more detail in the next couple slides. So frame shifts just refer to the order that we're looking at for the codons in the DNA. So we'll talk more about that in a second. But first, I just want to get at this idea of a silent mutation. So let's take this example sequence. We're looking at RNA right here because you can see the U's, and that's just because I want to use the codon wheel to get at this idea. But obviously, any change in the RNA would ultimately have been reflected in the DNA, right? So this change would be coming from the DNA itself. So let's look at an original sequence AUG UUC AGU. And hopefully at this point, you guys are pretty good at using the codon wheel or the codon chart to figure out, given a codon like this, what amino acid are we trying to, what amino acid are we coding for? And in this particular case, our three codons code for methionine, phenylalanine, and serine. So now let's imagine that this same DNA sequence that we used to get this RNA had a mutation. So when we translated that, or when we transcribed that DNA into RNA, we have a mutation that's popping up here. And instead of UUC, now for this middle codon, we have UUU. And you'll find that if you look, let's look at, let's look at this first one, UUC first. So UUC, and you'll see that that's coding for phenylalanine. But if you look at what UUU codes for, you'll see that it's still phenylalanine. So even though we have a mutation, the actual amino acid isn't changed. It's still phenylalanine regardless of which codon we use. And I think this will make sense because a lot of you recognized that for certain amino acids, like if you look at alanine, there are four different options for that would all lead you to alanine. If you have a G followed by a C, then it doesn't matter what the last letter of the codon is. It's going to give you alanine regardless. So because of this redundancy in the way that we code for different amino acids, we can get so-called silent mutations, or mutations where there's not actually a change in the resulting amino acid. So that would obviously be the best case scenario, right? A mutation that doesn't actually have an effect. So in this slide, we are going to be looking at what happens if a mutation isn't silent. So what happens if we actually do get a change in the DNA sequence that actually has an effect on the final amino acid sequence. Up top, you see our normal or unmutated situation. So we've got a DNA sequence, and then you can see what it looks like when it's transcribed into mRNA, and then what the resulting amino acid chain is going to look like. So first, let's look at a substitution where we're just going to be changing one nucleotide, one base. And you'll see that the change is happening right here. In the normal DNA strand, we have a C at this position, but it's been changed to a T instead. And because of that, in our mRNA, for this codon, instead of GAA, we've got AAA instead. And if you have a codon wheel or a codon chart handy, you can look this up yourself. But you will find that GAA calls for glutamine, whereas AAA calls for lysine. So we're actually going to get a difference in the amino acid that we're calling for based on this codon because of the mutation. But you'll also notice that before and after, everything else is normal. So we still start with a phenylalanine, then we have a tyrosine, and for the next codon, we're back on track. We still have a glutamine. So the change is sort of isolated to just that one codon that we messed up. And that's going to be important because when we think about insertions and deletions, sometimes that's not the case. Sometimes we actually affect more than just the codon that we're mutating. So in this case, it's an insertion. So we're going to be adding a base or more than one base. And in this case, we're just adding one. So before we had CTT, CTC. But here we're adding an extra C before we start that chain. So instead of CTT, CTC, now we're going to have CCT, and that's one codon, and then TCT. So everything is sort of like pushed back by one base. And so our mRNA codons are going to be completely different um, based on that insertion. So we have GGA, which is totally different than GAA. And then the subsequent codon is also going to be messed up. Instead of GAG, now we've got AGA. And that's because the way that we're grouping our nucleotides to make our codons is all off by one now. So now instead of glutamine, we've got glycine, but we've also messed up the following amino acid. We've got instead of glutamine, we've got arginine. So an insertion, if you add one or two bases, then you're going to totally alter the way that those codons are read. And so every subsequent codon is going to code for something different. Obviously, the effects of this are more detrimental than a simple substitution where we're only screwing up one of the bases or one of the amino acids. Um, and then the same thing for deletion. So here we're getting rid of the A that should be starting off this sequence. So instead of ATG, now we just have TG. And then the C from the start of the next codon gets grouped in with the T and G. And we're off by one. So with insertions and deletions, when you get a frame shift, you don't only mess up the codon that you're where the mutation occurred, but you also mess up every subsequent codon. You might be thinking, well, what if you add in 3 new nucleotides? So what if we put in 3 Cs here? And then that way, it would sort of keep the codon groupings the same. And that does happen. And those types of insertions and deletions, if you add 3 or you get rid of 3, tend to be less detrimental than this version where you screw up the grouping. And this is this example of when the codons become grouped incorrectly because there's an insertion or a deletion. That's the frame shift mutation that I was mentioning earlier. So a frame shift mutation is when you add or get rid of nucleotides, and then the codon grouping situation becomes all messed up. You're shifting the frame of like which codons should be grouped together. Okay, so we've just got a couple more slides. What causes mutations? There are two sort of categories of DNA mutations, and one we talk about as being spontaneous. And these are mutations that are just going to happen as DNA is replicated or as meiosis is happening. So anytime DNA is replicated for cell division, there's a chance that DNA polymerase is just going to make a mistake. And the cell does have sort of proofreaders to catch those mistakes and hopefully correct them. But there is just a certain low number that are going to escape notice and pass through. So those are spontaneous mutations, and there's nothing really you can do about it. That's just a baseline level for everyone. But there are also agents that can come in different forms that cause mutations. And we call these agents mutagens. So mutagens are just things that cause mutations to happen more frequently. And so if you think about things that people might think of, UV radiation. So like that's why tanning is bad for you because it damages your DNA and can cause mutations. And also carcinogens like cigarettes cause mutations as well. But there are also other things, X-rays. That's why they put that heavy like bib on you when you get an X-ray done. Processed foods, certain cleaning products, and there are actually viruses and bacteria that can cause mutations as well. So that's part of why HPV is so dangerous because it can actually cause mutations in cervical cells. Okay. And then last thing, I just want to talk about the effect of mutations. Are all mutations bad? I've kind of been talking about them like they are, but mutations are random. So when the DNA is mutated, it's not trying to hurt an organism. It's also not trying to help an organism. It's just totally random. And the effect of that mutation can be beneficial, it can be harmful, or it can be neutral. As it happens, we've had many, 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 many years for evolution to fine tune different ways for genes and proteins to be functional. So, in actuality, it's more likely for a mutation to break something rather than for a mutation to create something that is useful or neutral. So it's more often harmful, but it can be beneficial or neutral. And mutations are ultimately where all of the genetic variation comes from. So when you're thinking about things that come in different colors or different sizes or different shapes or work in different ways, that all comes down to a mutation that popped up that happened to be beneficial or at the very least neutral. Um, so so yeah, in that sense, mutations are pretty cool. Okay, that wraps up this PowerPoint, and I will talk to you all later.